All right, hey everybody. I hope you had a good first day at school yesterday, and that everything went well with this. I'll be checking in with Miss Schultz. 'Cause I'm recording this on Wednesday. It's Wednesday about 1 o'clock, so I'm recording this Wednesday for you guys on Thursday. And so I hope this is going well today. Again, I'll text Miss Schultz and see how everything's going, and I hope you guys are doing what we need to be doing, and this is going well. So this is for Thursday. What we're going to do today for biology is I'm going to make two videos. One short one on these dihybrid crosses. So one short one just on that, then I'll stop it, and then we'll pick it up. I'll start another one, and then we'll go into complex inheritance patterns, and that'll be mainly for Thursday and probably some on Friday as well. And so, but today it'll be maybe a little bit tougher than yesterday. Hopefully, that stuff went well. But a lot of the terminology that we'll use today is going to be the same thing we used yesterday, so it builds on itself. And so it gets tough if we're using terminology today that we don't quite understand. So you can always again pause the video, go back, and say, "Whoa, what does that word mean?" and check back, and then come back to it. Okay. All right. So let's get started. I'm going to present this, and we'll start right here again. This one won't be long. All right. Here we go. All right. So yesterday, let's go back. We're on the slide. All right. We had monohybrid cross. So monohybrid is one trait, right? We're talking about one trait. So this was the monohybrid cross, what we did yesterday. It's when we have one trait, say pea color, right? The color of the peas. And you'd say, okay, well, let's just say this pea, let's say big A stands for green and little a stands for yellow. Well, this is a dominant recessive. So we'd have a green plant crossed with a green plant, but they're both heterozygous, but it's just pea color. So it's one trait. All right. Well, that's a monohybrid cross. All right. One trait. Well, if we want to look at two traits at the same time, what about like pea color and pea shape at the same time, right? So like green or yellow or round and wrinkled at the same time. Okay. So we can do it with what we call a dihybrid cross. Okay. So this is a dihybrid cross. All right. So I'm going to write in here. See how this works. Okay. Yeah. So let's read it. All right. Dihybrid crosses are used when finding the possible genotypes for offspring when considering two traits at the same time. So example one. It says cross two tomato plants that are both heterozygous. So they're saying they're big T, little t. Okay, that's for tall, right? And they're big R, little r. So you could say the color, maybe like red and yellow. Okay, tall, short would be big T, little t. All right. So what are the genotypes of the parents? Well, mom, since they're both heterozygous, we don't really have any options here. So we know that it's going to be that mom is going to be big T, little t, and big R, little r. And dad's going to be the same, big T, little t, big R, ooh, and little r. Okay. All right. So it's big T, little t, big R, little r. Big T, little t, big R, little r. Okay. And that's what they are. It says now sort the alleles. It says ask yourself. This is where this gets kind of confusing. But it says ask yourself what combinations could go into the gametes during meiosis. Okay, man, two words right here. Gametes during meiosis. All right. So meiosis is the production of sex cells. Well, what are sex cells? Sperm and egg, right? Sperm and egg. Okay. So that's meiosis, the production of sperm and egg. And so the gametes are our sperm and egg, right? So what combinations could go into the gametes? All right, so we know that, or will, and again, we kind of skip meiosis, but we're going to go back to it. All right, but so anyway, during meiosis, you or your mom gives one version of a trait, and your dad gives one version of a trait, right? So you each have one, or so you have two, one from mom, one from dad. All right, we've said that. All right. All right. So in this case, we could have, so let's look at this one. We got big T, little t. All right. Big R, little r. All right. So that means that one of these, either big T or little t, could go into the sperm or egg cell, and big R or little r could go into the sperm or egg cell. Only one of them, not both. Okay. So let's say mom, in this case, is going to give a big T. All right. So this is going to be, in this case, we're going to draw here. That's our sperm cell. Okay. All right. So mom, I'm going to show you guys another way to do this in a minute. But anyway, let's just stay with me here. Okay. Dad's over here. I hadn't put his yet. So dad is the same. Big T, little t, big R, little r. All right. Well, what's the possibilities here? So let's say, okay, well, mom could give a big T and dad could give a big T. Okay, so it could be big T, big T. Well, mom could give a big R and dad could give a big R. So it could be big T, big R, big R, right? We could say that. That could be in this sperm cell, right? Well, she could give a little t, he could give a little t. I'm just going to talk through these here and I'll show you how to make more sense of this in a second. But mom could give a big T, dad could give a little t. Mom could give a big R, dad could give a little r. They could both give little t, little t, little r, little r. Okay, it could be homozygous recessive. Okay, you could have all those different combinations. So it's like, well, when we're making our Punnett square, the big thing is that when we're making a little square, how do we make sure we get all the possible combinations on the square? Okay, let me show you an easy way to do this. Okay, it's called the full method. Y'all know the full method from math, maybe. It goes first, F-O-I-L, F-O-I-L, okay? Full method. So the full method stands for first, outside, inside, last. So let me show you, okay? So what I mean is when we, on the next, we're going to see, maybe better do this on the next slide. Let me see here. Let's go back. Let's go to the next slide and see if yeah, this will be better on this slide, okay? All right, so let's do it on this slide. It says, write these combinations on the sides of the Punnett squares. And right here on number 3. In this case, we will use the same combinations on the top and the side because the mother and the father are the same genotype. So what it means, you only have to do the full method one time. Okay, so if you're lost, we're about to get it here, okay? I'm actually going to remove that real quick. Okay, so what we do, let's write down their genotypes again. Okay, so we only have to do it once because mom and dad are the same, right? They're big T, little t, big R, little r. Okay, so the full method, big T, little t, big R, little r. Okay, full method. First, outside, inside, last. Okay, so what we can do here is we can go first to, so that would be big T, big R. Okay, outside, these two. So on the outside, big T, little r. Inside, which would be little t, big R. And last, which would be little t, little r. Okay? And now, what we do is take these combinations, big T, big R, big T, little r, little t, big R, little t, little r, and put them on the Punnett square. Okay? And we can put them on both sides because it's like mom up here, dad down here. And we can go ahead and, it's the same though, right? Mom and dad have the same genotypes. So that we know it's the same. So what I can do here is do that. I can go big T, big R. Okay, and remember this, I knew this because of the full method. F-O-I-L. Okay, I did first, outside, inside, last. First two, big T, big R. Outside, so this is on the outside, that one's on the outside. Big T, little r. There it is. Inside, T, R, right there. Little t, big R. And last, little t, little r. Okay, so let's put them up here. Okay, all right, now we have big T, little r, big T, little t, big R, and little t, little r. This would be way easier with a pencil, guys, but I wonder, should have tried this out. No, that's okay. All right, so now we do the same thing over here, and I will go over this more in class. This is taking a little longer than I wanted, but nonetheless, because this will be on a test. You will have a dihybrid cross on a test, so we will have to do this. But this is not like the only focus, because this is a lot of work. It's a lot of work for one problem, but you'd have like one problem of these on a test, say. So okay, what you would do now is combine them, right? Big T, big T. These are all the possible gametes, all the possible sperm or egg cells that would come from this dihybrid cross. So big T, big T, big R, big R. Okay, and I would keep filling this in. Again, this is going to get really sloppy for me writing on here, but you would just keep doing this. Big T, big R, little r. And what you get is it's the same as doing a small Punnett square, just the 4 Punnett square, the 4 block Punnett square, except it's 16 when you have a dihybrid cross. Okay, the key to this, guys, the key to the dihybrid cross is getting the full method. Okay, first, outside, inside, last. Because to be able to fill out the Punnett square, you have to get the correct alleles, right? These are alleles, right? Different, remember the word allele, different versions of a trait, right? Because this may be tall. Big T stands for tall. Little t stands for short. Big R stands for round, say. Little r stands for wrinkled. You have to get the correct alleles on the sides of the Punnett square so we get the correct possible combinations in the square, right? And then this is just probability. Remember, it's like flipping a coin. When we fill these squares out, it'll tell us all of the possible combinations for our offspring, okay? And what you'll see when you'll do this is you get a characteristic. It's a 9, 3 to 3 to 1 ratio, which is like the classic dihybrid cross ratio. But so again, I'm not going to fill out this whole Punnett square today because we got a lot more to do. But I think if you get the point there, let's just do 2 more. Let me do one here. Let's do the last one. How about? So it'd be, what would it be here? This one here. This one, that's supposed to be a little t, right? Because I went big T, big R, big T, little t, big R. Yes. Let's see there, I'm confused myself. That's a little t, little r. Okay. So this last one would be little t, little r, little r. Okay. And that will be how you do that, right? Because I put that one there, that one there, that one there, that one there. Okay. And you just keep doing this. This one there, big R, little r, and just keep filling in the squares. And then you can get all the possible genotypes or letters ratios. And then you can figure out your phenotypes. Like this one would be tall and round. This one would be short and wrinkled. Okay. Okay. Cool. Okay. All right. And said number 4, it says combine the alleles from the top and left. That's what we did to fill in the square. Okay. Okay, then you can determine the ratio. All right. So let's do another example here. Okay, example, and then we're going to move through it relatively quickly. Okay. Put it over here. Okay. So it says, so this will give us an opportunity for the full method again. All right. In peas, purple flowers are dominant to white. Okay. And tall plants are dominant to short. A white short plant is crossed with a plant that is heterozygous for both traits. Okay, so the first thing it said we did, well, number 1 was we got to figure out dad and mom. Okay, so what are the genotypes of dad and mom? Okay, all right, so dad, mom. Okay, so right here, doesn't matter which one's dad, which one's mom. We gotta get it right for you gotta say, okay, dad's this one, mom's this one. All right, so here we go. In purple, in peas, purple flowers are dominant to white. Okay, so let's say, uh, what we just pick a letter, right? Let's say we could use P. Let's use. These are not really good letters. It's like good to have differences in the letters. Um, all right, so we'll use T for tall and short, but for let's use P, I guess. So big P is dominant. Little P is so big P will be purple. Little P will be white. And tall and short. So big T will be tall. Little T will be short. Okay, it's just a white and short plant. Okay, so we've got a little p, which I'm going to put a little, how about I put a little, for little p, I'm going to put a little cross through it, okay? We've got a little p crossed with a little t, okay? Okay? Because that's a white short, okay? Because white is recessive to purple, and white is and in short, so it's short recessive. Get this, guys. Okay, here we go. You gotta just keep your stuff together, and you got to keep it in order here. So we've got that. All right, so that's dad's. All right, and then it says a heterozygous. All right, so that's an easy one. So it's gonna be big P, little p. Okay, then heterozygous, so big T, little t. Okay, so now this one we have to do the full method twice. We have to do the full method twice because dad and mom are different. So this will be a little tougher because what goes on this side is different than what goes on this side because mom and dad have different phenotypes. But it's not the end of the world. You just have to do it twice. Okay. So, all right, I'm going to write it here for dad. All right, it's going to be little p, little p, little t, little t. Okay. But guys, look at the possible combinations here. What is the only possible combination that dad could give? All dad can give is a little p and a little t. So do you really have to do the full method? Try the full method out. All right. Well, first, uh, little p, little t. Outside, little p, little t. Inside, little p, little t. Last, little p, little t. So just put them all, right? That's it. That's it. So here we go. Little p, little t. Little p. I should put my thing. Little p, little t, little p, little t, little p, little t. Okay? All right. Here we go. So it's all the same. And now mom is going to be not all the same, right? So first would be big P, big T. Outside, big P, little t, big P, little t. Inside, little p, big T. And last, little p, little t. Okay? Then you'd put those. All right, little p, okay, little t. All right, we're getting this, guys. All right, little p, big T goes here, big P, little t, and big P, big T. Okay, so we got 1, 2, 3, 4, all the possible genotypes there. And then we do the same thing as we did before. We're going to go over this again in class. My writing's pretty slow, but hopefully this is getting somewhere, some decent examples from the full method, FOIL. So then we would combine, okay? This was mom. Mom can give a big P. Dad can only give a little. Okay? Mom can give a big T. Dad can only give a little. Okay? Same thing here. And if you'll notice, it's going to start to be the same thing over and over again. But let's go ahead and do it anyway. I'm not going to go through this whole thing, just to show you again. Okay, that is what you would do, and then you could go through and figure out your characteristics that you could possibly have in your offspring. Okay, these would be your alleles, right? The alleles on the edges. Then here are the possible genotypes for the children. Okay, so big P, little t, and you just keep going and filling it in until you have them all filled in. And then I could ask you a question. What's the ratio? We'll talk about ratios when we get there. I say, what's the ratio of and ratios just you could is just how many out of the total, right? So there's 16 possible total there. So how many out of the total? How many are going to be white and short? Okay, we could figure that out. We just count them up, right? You just count them up. Then you say, okay, it's 7 out of 16. Okay, because there's 16 total, however many it would be. Okay, you just count them up. All right, we'll do that when we practice, and you'll have some practice on your worksheet. Okay, all right, guys, we're going to stop there. I'm going to stop this video here. I'm going to upload it to YouTube. So this is some dihybrid cross practice. Like we said, the key is getting the full method and getting the correct things on each side of the Punnett square. You can do that. You got it. Okay, you can do 2 traits at the same time. Okay, I'll be back with another video